Hi, my name is Ishita Virmani and I'm currently a visiting scholar with the Center of Alternatives to Animal Testing in Johns Hopkins Bloomberg School of Public Health. I am a current fellowship recipient of International Foundation for Ethical Research. I would like to thank IFER for supporting my project entitled A 3D Human iPSC-derived brain organoids as a model for developmental neurotoxicity assessment of man-made chemicals. I would also like to thank Dr. Helena Hogberg, Dr. Eva Somadinova, and Dr. Thomas Hartog. Now, let's talk about the project. To provide you with some background information, there has been a rising concern about increase in developmental neurodisorders such as autism spectrum disorder, attention deficit hyperactivity disorder, lowered IQ, as well as learning disabilities. However, only approximately 200 numbers of chemicals were tested for their developmental neurotoxicity potential till date. This indicates a massive gap in data and knowledge. One of the reasons behind this gap is the use of animal models for the chemical testing. These animal testing methods use a large number of pups, are really expensive and as well as time consuming. Due to these reasons, there is a need of alternative approaches for the testing of chemicals. My project particularly aims to assess the developmental neurotoxic potential of a class of man-made chemicals called as flame retardants. As we all know that the flame retardants are added to consumer products such as electronics, furniture, and children's toys to delay fire propagation. These chemicals have been detected in school, offices, as well as apartments. The childhood exposure to these chemicals is of a particular concern due to their increased hand-to-mouth activity as well as their vicinity to the dust. Therefore, there is a need of testing these chemicals using models that are relevant to humans. In this project, I will be assessing the developmental neurotoxic potential of these chemicals using a stem cell-based 3D in vitro brain spheres. The brain sphere exposed to the flame retardants will be collected and used for measuring several endpoints such as cytotoxicity, the alteration in gene expression, and as well as the structural changes in these cells. I am currently performing the qPCR analysis to study the alteration in gene expression of the genes that are responsible for key neurodevelopment processes. These models for chemical testing have the potential to reduce, refine, and replace the currently used models for chemical testing. This project is very different and exciting from what I have done so far in my PhD. I'm enrolled as a PhD researcher at Resitox, Masaryk University in Czech Republic. So far, I have worked with the 2D in vitro models for assessing the reproductive toxicity potential of organochlorine mixture under the supervision of Dr. Eva Somadinova. Dr. Eva Somadinova has consistently supported me to gain practical experience and knowledge about the in vitro approaches for chemical testing such as 3D human brain sphere models. In the summer school for alternative approaches that I attended in June 2020, I attended a talk by Dr. Helena Hogberg and I also attended a lab tour of the CAT. At that point, I decided that I wanted to join CAT to learn more about the alternative approaches for developmental neurotoxicity assessment, and then I came to the CAT for 1 year. Doing this project at CAT will provide me with the new future avenues as well as broaden my knowledge about the developmental neurotoxicity.